Okay, so next we have Professor Rob Mullins from the University of Iowa, who's going to talk to us about his favorite topic, the choroid.
Well, thanks so much. And yeah, thank you, Sarah and Florian, for inviting me. Thanks, BrightFocus, for putting on this terrific set of presentations today. So I don't have any financial disclosures. I will disclose that I realize I am the only thing standing between you and lunch, but I hope that your grumbling stomachs will not get in the way of learning what will hopefully be some useful information this morning. So today I'd like to talk about ocular vasculature and especially focus on choriocapillaris and choroid, talk a little bit about some of the other cells that reside in the choroid. I want to focus for a while on the anatomical and molecular changes that happen during aging in the eye, and these include drusen, which we've already heard about, a little bit how age and disease and genotype all affect some of these molecular and anatomical changes, the role of monomeric CRP and complement, and then we'll talk about a few sort of new areas, pitfalls and advice, and unanswered questions. So one thing everybody should know if you're unfamiliar with this topic is the retina has two different blood supplies. And so the retina has its own vasculature, which is supplied from the central retinal artery and comes up through along through the optic nerve and spreads out and nourishes what's called the inner retina, whereas the outer retina is supplied by the choroid. And so this is just a cartoon with a illustration of the inner retinal supply, again from the central retinal artery. And the photoreceptors and RPE, which are cells that most people in this room are probably most interested in, are supplied by the choroid with its capillary bed, the choriocapillaris. Again, choroid, unlike retina, the arterial supply comes along through the ophthalmic artery, splits off into a set of ciliary arteries that penetrate the sclera and supply the choriocapillaris in that way. The choroid has anatomical layers that have been described for it. So there's a choriocapillaris, which are these very large bore capillary beds, and then a layer called Sattler's layer of intermediate vessels, and a layer called Haller's layer of large vessels. And so actually some animal eyes don't have these three different layers. This is really pretty much just primates and pretty much just in the macula. There was some really important seminal work done by Sohan Hayray in the 1970s in which he mapped out the supply of the choriocapillaris. And he did this in a way, I'm not sure we could do this today exactly, but he injected fluorescein into the carotid artery of monkeys and then did high-speed photography capturing the filling of the choriocapillaris and its drainage. And so what he found is that initially there's delivery through arterioles at the center of each choriocapillaris lobule, followed by filling of the choriocapillaris, and then drainage around the edges. And so this is still our model of how choriocapillaris is supplied. A little physiology of capillaries, in case you're not familiar with this. There are different morphologies of capillaries in different organs in the body. And so in liver, the most leaky, and in bone marrow, the most leaky are called sinusoidal. They have a discontinuous basement membrane with giant gaps between the endothelial cells that allows them to dump large amounts of albumin and other liver proteins into the circulation. There are continuous capillaries, which we're going to hear more about later today, that are in the retina and brain and where there's a very tight regulation of what's allowed in and out of the vessel. And then this intermediate kind of capillary is called fenestrated endothelium, and that's what's seen in the choroid and in the kidney. So choriocapillaris has what are called fenestrae, and this is in Romance language is the word window. Right? So the choriocapillaris has windows, and you can kind of appreciate these little windows where the endothelium looks sort of pinched. And so they're mostly oriented toward the RPE, and they have diaphragms in the choriocapillaris that's different than kidney, where they're open. And they can be induced in vitro with agents like VEGF, and they can be suppressed by agents like anti-VEGF. And I think we'll hear more later about endothelial fenestrae from Dr. Demore. Now, these fenestrae, again, this means window, And I always thought that in aging there was loss of choriocapillaris fenestrae, but it looks like that's not exactly the case. There's this study done by Jerry Luddy in which they quantified the number of fenestrae in choriocapillaris endothelial cells, and between young controls and aged controls, there wasn't a large difference. The language for the genetics is a little different. What was described as pure 1 meant that these individuals had a high risk for complement factor H genotype, and a low risk at the 10q26 ARMS2HRA1 locus, whereas pure 10 was just the opposite, so low risk for factor H, high risk for ARMS2HRA1. And these what they called pure 10 samples had reduced numbers of fenestrae in human eyes. So this hasn't really been followed up on. It's very interesting, though, when we think about, as Ray was talking about earlier, how do these genetic variants cause disease? So this is a process of defenestration in the endothelial cells, and it's quite different to the defenestration that we're all going to be learning about as we learn about the history of Prague and the defenestrations where the Habsburgs were chucked out of the windows. And so choriocapillaris is it's a dense, large capillary bed. Again, the cells are fenestrate. It supplies the outer retina, and you can appreciate in a whole mount like this just how profoundly dense the vasculature is. All of the little dark spaces is actually where there's not an endothelial cell. All the red is where there is. So it's more than 50% of the cross-sectional area is actually vascular lumen. In addition to endothelial cells in the choriocapillaris, there are pericytes. So these are the cells here. And they have some interesting features. They play a supportive role for the choriocapillaris endothelial cells. They have structures like these hemidesmosomes, which they adhere to the matrix. They share a basal lamina with the endothelium, and they have some particular markers which you can also use to follow them on single cell studies. And so there are numerous cells of the choroid. We were learning more about these with the kind of work that Ray was talking about earlier with single cell RNA sequencing, and we can identify multiple different populations across the choroid that are of different interest to different folks in the audience. In terms of the structure, we can appreciate this a little through serial block face EM. And so if this works, this will be amazing. OK, great. So we're sectioning through the RPE. And you can see the lipofuscin granules as we cut in. And here's Brooks membrane that we're getting down into. And where you start to see these little like lightning bolts, that's the elastic lamina. All right, but here's the lumen of the choriocapillaris. And you can see that Brooks membrane pinches, oh, there's a red blood cell. You can see that Brooks membrane pinches off into these intercapillary pillars. But look at all of this lumen. So there's just, again, an enormous amount of space within that layer. And now as we exit the bottom of the choriocapillaris into the choroidal stroma, you see abundant collagen, as well as this draining venule that will follow the rest of the way. And so the choriocapillaris anatomy, again, is just, it's striking how dense it is under normal circumstances. So what happens during aging? And so there are a number of structural changes that we can talk about, as well as molecular ones. Structurally, we've heard already about drusen, and that these can be called hard or soft. And this is what they look like clinically and what they look like histologically. And so histologically, hard drusen, part of the reason why I think that they were easy to see and why they were called hard clinically is because when you're looking down through the pigment sort of piles up on either side of these little nodular drusen, and so they're well highlighted. Whereas soft drusen, the pigment is sort of stretched out, over these more extensive deposits. The other main important for AMD histological deposit is this material called basal laminar deposit. And when there's a confluent layer of basal laminar deposit, this is what Sarks and others used as the histological definition of AMD. So I think, when you're thinking about animal models, you won't see generally a lot of big drusen. the deposits usually in animals or in mice will be less than the height of an RPE cell. But you may see something more resembling basal laminar deposit like this. Also very important for AMD. Hard to see clinically unless it becomes detached, in which case this resembles soft drusen. And clinicopathologic studies of eyes that had soft drusen that were subsequently studied by Dick Green show that this is a frequent manifestation of soft drusen, detached basal laminar deposits. So what are drusen composed of? We know that they're about 40% lipid. The remainder is mostly glycoproteins. It includes things like complement components, MHC antigens. There's a lot of TIMP3, ApoE, and some other glycoproteins. The composition of basal laminar deposit is not as well appreciated, and maybe it's collagen 6. It has a cross this sort of railroad track EM appearance of repeating collagens that's similar to what's called long spacing collagen. It's not really clear though what it's made of, even though it's very important. So it's an important area to keep exploring. All right, so in addition to deposits, aging eyes have choroidal thinning. And so we lose about 15.6 microns per decade was the original study. This can be studied in the clinic with OCT now and can be measured. And so every year, we lose 1 to 2 microns of choroid. So if you come back to the RD meeting in 2027, your choroid will be 2 or 3 microns thinner than it is today. Now, in some cases, though, so there's normal aging, there's choroidal thinning. In some pathologies, there can be dramatic changes in the thickness of the choroid. And this is also an area that there's not much understood in terms of what the molecular basis is of this. Is it just vascular attenuation? Is it something else? But this is what it looks like in a large study of 9,000 individuals. The x-axis is different decades of life for men and women. And you can see this notable just loss of choroid. In males, there's also a loss of choroidal density of the vasculature. So the proportion of blood vessel area to the choroidal stroma area was reduced in males, but not females in this study. There are other things that can affect the choroidal thickness in sort of in small ways. So if you consume a large quantity of liquids and increase blood volume, you can incrementally increase the choroidal thickness, but it's small. It changes posture, it changes a little bit. Biologically, things like depletion of macrophages results in choroidal thinning. And this was a study by Wai Wong, and it looks like a typo or something. What do you mean deplete macrophages, you thin the choroid? But somehow, when macrophages are present, they're keeping the choroid at a certain volume, which is reduced when they're gone. And when they're replaced, it grows back. Microgravity, astronauts coming back to Earth have a thicker choroid than they had when they left. And it takes weeks for it to resume. There are concepts for why that is. We're not really sure. A recent study showed that circulating concentration of C-reactive protein was related to choroidal thickness. So again, tying in some of the work we've heard earlier in terms of role of inflammation and macular degeneration. And then in disease, we see changes. Central serous retinopathy, or central serous chorioretinopathy, choroid is much thicker than in controls. Geographic atrophy, it's attenuated. And this was a donor eye study we did. The problem with doing this in donor eyes is there's no blood pressure anymore pumping up the choroid, but in the absence of blood pressure, we see that in geographic atrophy, there's significant loss of choroidal thickness. Well, in addition to loss of just the mass of the choroid through choroidal thinning, there's also local loss of cells that occurs. And this is particularly noted in the choriocapillaris. And so if you look at a cross-section like this of an eye with early AMD, you can see intact RPE. Now, when I showed this slide to Pat DeMore, she said, yeah, that RPE looks like a train wreck. I was like, okay, it doesn't look the greatest. But look at the choriocapillaris. So the red vessels are living vessels. There's exactly 2 choriocapillaris vessels that are alive in this section. And these dark vessels where there's a lumen but no endothelial lining, or what have been referred to as ghost vessels. So it's a remnant of where a blood vessel used to be, like a shed snakeskin or something, but there's not a living endothelial cell in it. It's not conducive to blood flow. And when among the many functions of endothelial cells, one is keeping the blood in a liquid state. And so when blood runs into the subendothelial matrix, it'll immediately coagulate. So there's no flow through these ghost vessels. So we did an experiment where we wanted to quantify these, and we had 147 eyes. We only studied the ghost vessels when there was intact RPE on top of them, and we measured them in a masked fashion, counted the number per unit length of Brooks membrane so we can standardize, because sometimes a section is long, sometimes it's short. And when we did this, what we found is that in going from age-matched controls to early or intermediate AMD, there's a significant increase in the number of dead blood vessels per unit length. But in geographic atrophy, when there's still intact RPE, so in the center of the atrophy, we expect there to be no blood vessels because RPE are needed to keep endothelial cells alive. Outside of the atrophy, there was a striking increase in the number of ghost blood vessels. We also saw this in a different study where we quantified the density of drusen and related that to the number of ghost vessels and found a linear relationship between abundance of drusen and number of dead blood vessels. And so this was all histological work. And then subsequently, as OCT angiography came on board, and there was much better ability to see past the black paint of the RPE in the clinic, there were subsequently a number of clinical studies that showed, yes, there are striking choroidal defects that occur in macular degeneration. These have been ignored, mostly because of the technology, and now it's becoming clear that this is an important feature of the disease. There are also some molecular changes that happen in the aging eye, and so one of the most interesting of these is lipid accumulation. It's not something that my lab studies, but there's accumulation of neutral lipids and phospholipids throughout age, this is what Christine Curcio has described as the lipid wall as a barrier for things like retinoids getting across from the vasculature to the RPE and a diffusion barrier. Another change that happens in aging choroid is the accumulation of C-reactive protein. Now, C-reactive protein, you may know, is a protein that's made entirely in the liver. It does not have an ocular source as far as we can tell. It's made in the liver. It circulates around as a pentamer. And when it encounters a dead cell or some activating surface, the pentamer falls apart into monomeric CRP, which is insoluble and is pro-inflammatory. And so the ratio between the monomeric and the pentameric form of CRP is important in terms of inflammation and what's happening locally in tissues. And so one thing that happens during aging is there's an increase in deposition of CRP here in around the choriocapillaris. More striking than aging, though, is this phenomenon occurs in eyes with high-risk factor H genotypes more than in low-risk factor H genotypes. I mean, and it seems to be specifically for the Y402H is the variant that's been studied. And so you may know that the polymorphism, the Y402H polymorphism in complement factor H gene is in a CRP and glycosaminoglycan binding domain. If you've got that variant, you get more CRP in the choroid. What's it doing exactly? Probably something interesting. And there's a recent paper by the Mullins group, no relation, studying C-reactive protein and driving neovascularization in animal models. Well, the aging change that we've studied most in my lab is the accumulation of membrane attack complex in the choroid. And so this is a cartoon and an amazing atomic force microscope image of what the membrane attack complex, or the MAC, looks like. And so there's a C5b, a C6, a C7, a C8, and then this ring of C9s that form the membrane-spanning pore. And the reason that we have the MAC isn't to give us AMD, it's to kill invading pathogens. But like with many things in biology, something that's supposed to be helpful can be harmful under the wrong circumstances. So where factor H fits into this, or where the MAC fits into this pathway, and there won't be a test about this or anything later, but there are multiple pathways for initiating and moving complement along to the final formation of the membrane attack complex if it's not stopped. And I'll show you this figure again a little bit later. This is where CFH fits in, is inhibiting C3b and acting as a cofactor for factor I. And but however complement is activated, if it's not stopped, it'll result in the formation of the MAC. Now, when we use antibodies, monoclonal antibodies directed against the MAC complex, and this is an antibody that reacts with the neoepitope on the C9 protein when it folds and inserts in the membrane, what we see is that MAC in the posterior pole is deposited in the choriocapillaris. And so it's not much in the neural retina. It's not much in the RPE. It's abundantly deposited in choriocapillaris. The same layer that I was showing you earlier is the layer where cells die under intact RPE cells in geographic atrophy. And if you're able to stereo this, this is a confocal stereo pair of green endothelial cell with red MAC on its surface. And if you can't find me later, and we'll work at it together, because this is worth learning to do if you haven't yet. Used to be, I have limited time. Okay, used to be you'd bring two projectors to a meeting like this, and one would be left eye, one would be right eye, and stereo pairs. People were very used to it, but that was the old days. my choroid was thicker back then. So we also use the same antibodies for ELISA to biochemically measure how much membrane attack complex is present in different categories of choroids. So this experiment, we had 10 young eyes, younger than 40, compared to 10 older eyes, greater than 80 years of age. And we quantified the abundance of MAC biochemically, and you can see during normal aging, there's about a doubling of the abundance of the MAC that you can detect. If we add in eyes with AMD, this is doubled again on top of normal aging. So here is an insult that can potentially kill cells that is driven by the high-risk genotypes, or one of the genotypes we know about, and shows this profound increase along with aging and AMD. And one of the things we do notice is that we want to understand the difference between normal aging and disease, but the difference isn't always that great. Sometimes things seem to be in the same direction. And so comparing a young eye to an old eye with AMD, the delta between these is not always so great. So I think understanding aging may actually be terribly important in understanding age-related diseases. Maybe that's obvious. All right, so one of the things we were charged by BrightFocus to do, we had sort of a rubric to fill out for these different areas. And so I think I failed abominably, but we'll see. So one of the things, one of the areas was new insights and things we're learning. So really did a great job of talking about the gene expression, single cell data sets that have come along. This was an experiment we did where we were comparing old and young we were single cell RNA sequencing, we're comparing old and young cells. And one thing that was striking, and some of this is published, some of it isn't yet, is that genes that are involved in inhibiting complement activation show decreased expression in older endothelial cells. So, and it was really, the cast of characters on this on this pathway. So all these genes in red, SERPING1, CFH, CFI, CD55, CD59, clusterin, all show decreased expression by choriocapillaris in old compared to young cells. So during normal aging, our ability to fend off complement in the endothelium is decreasing. I do want to point out a lot of this data is on Spectacle. So if you haven't used this website yet, please do. It's we have multiple different data sets from different papers loaded in there. You go find the paper you want to interrogate and type in your favorite gene name, and it'll show you what cells express that gene. One other new area that we had nothing to do with in my lab, but I think is very important for choroid, is there's a recent paper out of New Mexico about the ability of choriocapillaris pericytes to detect light and respond independent of any photoreceptors being in the area. So this isn't something where photoreceptors do some signaling to the RPE, does some signaling to the pericytes. This is a pericyte-independent opsin expression that drives contractility by light. So very cool area. So to summarize, we see that human choroid has structural and molecular changes during aging. Some of the same changes are present in AMD, just to a greater extent. These changes can be a cause or maybe an effect of a pro-inflammatory milieu that includes things like complement and C-reactive protein. And several of the molecular changes that we observe in eyes and the anatomical changes are related to the genotype of the individual. So high-risk genotypes drive some of these changes. We see age-related complement insult along with age-related loss of anti-complement defenses. And again, aging and AMD are really quite similar. I wish we're still looking for the magic bullet to disentangle those. So just in sort of graph format, we see loss of factor H, CD55, clusterin, other inhibitors of complement at the same time that the MAC and ICAM1 and CRP and oxidative stress are going up. But this delta is even greater in the presence of the Y402H genotype. So all of the things we see in normal aging are worse in AMD and worse if you have Y402H. All right, so one of the things we were asked to do is have some take-home messages. So these may or may not have anything to do with choroid, but anyway, there are things that if I was speaking to a group of people at different stages of career, maybe trainees, maybe not, but these are things I'd want to communicate. So if you look at the person next to you, sitting next to you, if you're not in the same lab, maybe even if you are, you have different abbreviations that you use to describe things. And so if you're writing a grant application to BrightFocus or anywhere else, and you have some weird abbreviation, PCR, okay, you don't need to spell it out. If you have some abbreviation that's unfamiliar, you should try to avoid using that abbreviation. It's like, well, I already told you on page 1 what it is. It's like, okay, but I'm on page 7 now. I forgot. I'm old, So, but to the extent you can make things easier on a reviewer by not bogging them down with a bunch of like your lab-specific lingo, I think that's better. Another thing, people will say, well, neovascular AMD is only 10% or something like that. The rest of AMD is dry. That's geographic atrophy. You should know geographic atrophy is a subset of dry AMD. It's not all dry AMD, and that's the kind of thing that if you put that in an application or paper, say, oh, that person's naive. They don't know what they're talking about. You don't want that. So be aware of that stuff. I'd also say, in terms of the genetics, whether it's mice or human, you want to be specific. If you're ruling out genetic risk factors or ruling them in, we know about RD8 in mouse. You have to do that, otherwise, there's a certain reflex in reviewers that if you have a mouse grant and you don't say that you looked for RD8, it just activates this this reflex that you don't want activated. Same with AMD risk loci. Say which ones, what you're going to do with the data when you get it. Be aware of the histopathology. So Christine Curcio has done all this fabulous work on the order that photoreceptors die. And, somebody will have like a cone degeneration model and for a mouse or something, say, hey, that's like AMD because the macula has the cones, and the cones died, and so this is like AMD. And whereas, Christine has shown, well, the rods actually die much earlier in AMD than the cones do, and this is something else. And so, so I guess, all that to say, be aware of the literature for those kinds of things. If you're a biologist, talk to a clinician. If you're a clinician, talk to a biologist. If you're a clinician scientist, you probably already talked to yourself. But anyway, but get input from your peers because for me as a basic scientist, I know that somebody like Dr. Chakravarthy knows all this stuff I don't know, and so that can be it can help you navigate these areas. Also, I'd say you don't want to do it alone. It's a hard job, and you want people around you that are going to help you carry the load. And colleagues can make our science better. It can also help keep us sane. All right. So a couple more things. Best models to use. We were asked about this. I mean, I don't know. It's tough, for macular degeneration. I think there are all kinds of for the BrightFocus review panel. There are 20 scientists on there. Something there are probably 20 different answers to these kinds of questions. I think you can use ARPE19 cells. You can use mice that don't have a macula. You can use iPS cells that don't that are immature and everything. I mean, we have to use these models because this is otherwise the only way not to use these is not to do anything, and that's not the right answer. So I think to me it's the issue is that you want to accept the limitations of your model, and you want to be thoughtful about it and show the reviewer that you've thought about it. But so mice don't have a macula; they have a Brooks membrane. Well, I'm studying Brooks membrane. I want to learn about Brooks membrane with my mouse model. That's a legitimate approach, I think. I've never written that mice don't have a macula review, by the way, in 50 review panels I've been on. So anyway, I think accept the limitations of the model. You don't want to sort of hide it or something. But you can't use a model to explore the totality of disease. You're exploring a part of it, and I think that's legitimate. All right, unanswered and future questions. As we've heard earlier today, we're really good at discovering genetic risks and altered transcripts. We're less good at figuring out what they do. Oh, and this, I guess, one more thing to the previous point. We all do this. There's a big list of 30 or 50 AMD risk factors. We look down the list of genes and we say, oh, that's interesting to me. I study this. A thing to know though is a lot of those SNPs might be 100 kilobases away from where the gene is that we're calling that's the collagen 8 SNP. It's not a collagen 8 SNP really, but I mean that was the nearest gene. But you shouldn't say, well, I study collagen 8, so here's this collagen 8 SNP. This makes my work important. Your work's already important, but anyway, I think to be aware of those, that we use a shorthand, and reviewers and others, and yourself should be, aware of those. All right, so we're good at discovering genetic risks, less good at figuring out what they do. So this is an important future area. As Usha and Kieran said, is there a tipping point for these diseases? Why do the cells decide to become atrophic? Why do they decide to? Why do blood vessels decide to erupt through Brooks? Can we find out what those things are? What's healthy aging? And there are all these other cells in the choroid. What are they doing? And with that, I did made a list of references for future reading. This will be on the video, so we'll all be able to look at these and sort of some historical, some new things that I think are important. And at that, I've got some time left, so we're going to not be as late to lunch as we might be, but otherwise, thanks very much for your attention.
Thank you very much, Rob. We'll
Thank you.
go to the room with some questions.
Okay, you'll have that going.
I hope you have it going in the background.
Yeah. The tech guys could make it on loop, but wait, next. There it goes. All right. Yes.
Do you want me to go ahead? Oh, right, okay.
The video is so beautiful, nobody wants to talk over it. But
So, Isha Chakrabarti again from the UK. Rob, that was just an incredible description of the choroid and its role. So, essentially, I just want to follow up with the question of nomenclature. And we, the clinicians, have been horribly guilty of muddying the nomenclature. So when people stand up and talk about dry AMD, sometimes they also think that after you treat choroidal neovascularization and the macula becomes dry, is that also dry AMD? So patients then come to me and say, have I now got dry AMD? So it's frightfully confusing. And I do think that we need a unified language to describe what we have. So, and suddenly I'm going to contradict exactly what I said in my earlier talk in some ways by saying that we probably shouldn't be calling drusen AMD, because would you call carcinoma in situ cancer? You just wouldn't, would you? So, and to me, macular degeneration should really reflect that huge degenerative process, which is when you have a huge patch of geographic atrophy or neovascular AMD, then you can tell a patient that. So coming from a clinician perspective, I mean, I totally take home exactly what you've said, and using the term dry AMD can mean somebody with just little, maybe minute little drusen to next to nothing. So well said.
Yeah. Well, thank you. And we used to have this language, ARM, you know, age-related maculopathy, but I think it's kind of, I don't think anybody uses that anymore. So. Yeah.
Ernst Hamm from University of Regensburg in Germany. You mentioned at the beginning of your talk that there are ganglion cells in the choroid, and as I recall, it's about the same number as in the ciliary ganglion, so there are quite a huge number of cells in the choroid. So my question would be, are there changes in thickness of the choroid that you can measure as a clinician? How dynamic are those? I mean, could those cells increase blood flow, make the choroid thicker or thinner, you know?
Yeah, thank you. So, yeah, you know, the regulation of choroidal blood flow, which is less auto-regulated compared to retina, but, you know, there's new data that there's some auto-regulation. You know, it's interesting. Folks, Anton Reiner and Melinda Fitzgerald had mapped the innervation of the choroid through the tracer studies with HRP and everything, and they trace back to these nuclei in the brain that I can't remember what they're called right now. But they lesion those nuclei, and they saw choroidal thinning, and you know, if I remember that right, they saw sort of deposits in Bruch's membrane, and it's not something that's been terribly followed up on. But you know, so that's not the resident neurons, the choroidal ganglion cells. This is you know, innervation from the brain through the cranial nerves. But yeah, I mean, those cells, exactly what they're doing and how they're maintaining choroidal health is very understudied. Thank you.
Hi. It's Andrea Barbino from National Eye Institute, and a terrific summary of choroid. Very good talk. My question is about if is known or there is an idea of if there is any causality or timeline between the appearance of ghost vessel, the deposition like of the thinning of the choroid, and the deposition of phospholipids or natural lipids.
Yeah, thank you. I don't think that those things have all really been studied in the same samples in the same way, which makes it difficult. I would say, you know, the choroid capillaris and the choroid overall are not terribly well correlated. So, you know, eyes with myopia, you know, a healthy eye with, otherwise healthy eye with myopia may have a pretty normal choriocapillaris, with a very thin choroid otherwise. So these terms aren't tightly connected to each other anyway. Thank you. Jason.
Maybe this is a slight follow-up on that. Choroidal health, as we get older, as you pointed out, includes this loss of fenestration and ghost vessels. How unique are those two processes from each other? And do you think separate pathology, well, obviously you think complement drives the ghost vessels, but does complement drive loss of fenestration, or is fenestration all about loss of anti-VEGF support? How much are those pathways totally together, in just different phases of the same thing, or totally different pathomechanisms? Yeah,
thanks. I mean, the fenestrate part, we haven't studied or thought about very much. It's hard because you need to do EM, and you need EM on a lot of samples, which is what Jerry had. And then especially for rare risk variants, like relatively rare, like 10q26, that's a pretty small fraction. But yeah, I mean, I think I would say that the loss of fenestrae is an RPE failure to support the choroid capillaris, and maybe that's also the cause of the ghost vessels. We don't know, but complement is also a strong contender for that.
Rob, kind of a continuation of Jason's question. Would you, based on the data you showed today, would you allude that there's an independent disease happening in the choroid as AMD is progressing, or do you still think that a lot of this is RPE, Brooks membrane interface dependent phenomena that is happening in the choroid?
I'm not totally sure I understand the question.
So based on what you showed today, would you argue that there's an independent disease progressing in the choroid as we talk about AMD, or do you think the processes are still coming from RPE, Brooks membrane area?
Yeah, thank you. So yeah, I mean, that's obviously a big question in the field, is where the disease originates. And I mean, our work of loss of choroid capillaris associated with AMD when there's still RPE there would argue, I think, that the damage is initiating in the choroid. Now, is it possible that RPE isn't doing something it needs to do? So even though the RPE is still surviving, RPE atrophy is clearly a late part of this disease, in my view. In fact, you can have RPE sitting on top of an old fibrovascular scar for years. They don't care. It's a robust cell. But if it's failing to do its job in maintaining the rest of the health of the eye, then that could have these other consequences. But yeah, no, from our work, I would say we have a choriocapillaris-centric view of the universe in terms of AMD.
I wanted to point out these dementor-looking cells that are flying in your choroid, which are, I think, the fibroblasts. We have some evidence that fibroblasts are very active in the choroid. Fibroblasts are very active in gene expression in maintaining the health of choriocapillaries and also immune response and complement pathway, perhaps. So I wanted to ask, what are you, and I think Rui's data also suggests that fibroblasts in gene expression are very active. What are your thoughts, or do you have any evidence in support of that fibroblasts are changing with age or with the AMD disease processes?
Sorry, where's the fibroblast?
Oh, that's mostly collagen, but there are plenty of fibroblasts. I think here comes one now. Maybe it's a macrophage in the bottom there. But yeah, so there are there's certainly a lot of fibroblasts in the choroid, and as anybody knows who tries to do primary culture of endothelial cells, because there's unwanted contaminant. Yeah, I mean, there are certainly age-related and AMD-related changes that are present. We haven't studied them as well. They're in our data sets. And on probably anybody from our 2022 paper data could look at that that has a laptop too. So, thanks.
One last question.
It's here. What happens to the diurnal variation in choroidal thickness in AMD?
So that's a great question. I know that posture has been studied, and there's some differences in choroidal thickness in early AMD patients compared to controls. I don't know if the diurnal changes have been studied, or if that's something that still needs to be done. Thanks.
Okay, thank you very much, Rob.
Thank you.